So thank you. That was great. It was great set of talks this morning, and especially a good introduction and examples from Victor and Jennifer about what the virtual brain is and what it currently offers and where we're headed. So I don't know that I need to motivate too much why we built the Macaque brain for TVB or for the virtual brain. But what I was hoping to do today, because of this opportunity we have for probably bringing this virtual brain and macaque brain to multi-scale models, is to describe how we created the macaque connectome for the virtual brain, the reasoning behind some of the choices we made to try to get a realistic representation of the macaque brain in TVB. And so let's get started. I want to be able to see everyone as well. I actually don't think I can. So if you have questions, feel free to just interrupt me as I go, because I want to make sure that this is clear. So when we started down this road of trying to create a structural connectome, so a representation of the anatomical wiring of the macaque, we were in a place where we were very privileged to have the choice between two different types of data to build that connectome. One is using tracer data, and where there's a ton of data already existing, and using anterograde and retrograde tracers in macaque to describe the cortical connectivity of the macaque brain. And then on the other hand, in the last, I would say 5 or 10 years, there's been a lot more neuroimaging done on the macaque brain, and a lot of diffusion weighted imaging that's been available so that we could run, for example, tractography to recreate the structural connectome. And in terms of large-scale modeling of the macaque brain, I would say most studies have used tracer connectomes, and we've learned a lot from these studies, and based on these tracer connectomes, a lot of those connectomes are built from the COCOMAC database, which is a very nice collation of tracer studies in the macaque. So here's one example of a study where the large-scale model was done to use to simulate resting state functional MRI in the macaque brain. And, I would say, and considering whether we should be using these tracer connectomes, they have very high accuracy because of the technique and what it does. We have good sensitivity and specificity of where these connections are and where they, for example, one region projects to another, or whether it does not. That the tracer connectomes are directed, right? We have the directionality of the axons and whether they go in from one area to another or back in the other direction, which we cannot get from neuroimaging data. And then, of course, the real advantage of these tracer connectomes is often that they can be integrated with other anatomical data. And I think part of the discussion in the last talk is anatomical, more data about, for example, the distribution of delays from all those afferents, or other ways in which we can build in that empirical data information that we already have into our models. So here's one example where this is a multi-scale model of the macaque brain, and specific to the visual system. At the large scale, they use this COCOMAC connectivity matrix from COCOMAC, which is binary, but they include layer-specific information about how those interareal projections actually connect. So, in this cartoon, it shows the very classical projection scheme of supergranular cells, and especially also deeper layer 5-6 cells projecting mostly to layer 4 neurons. And so, that's one example of how additional data can be used to enhance a connectome. But there are, I would say, disadvantages to this to this type of data. One is generally we have quite limited coverage. Historically, I'm sure most of you probably already know this, that tracer studies have generally been limited to single hemisphere tracing. So there's not a ton of information about how these projections cross hemispheres. And there are exceptions to this, and I'll show you one. And then, you know, certain systems have been really overrepresented in the sampling of these tracer studies. So for example, the visual system is really, its connectivity is really well understood, but there are other regions and other systems where information is still lacking, and it could be better. Right. And then in general, most of these tracer connectomes lack any information about what I'll call connection weights. So anything about the number or proportion of axons that project between one region and another. And so, you know, with that in mind, you know, we come to the other type of data we could have used, which is macaque tractography data from diffusion weighted imaging. You know, and we've seen a lot of these images already today where you can build these really beautiful and detailed pictures of the anatomical wiring of the brain. And, you know, the advantages of using those instead are that we have some estimate of the connection weights from the tractography, and then we have a very good estimate of track length, which is, you know, how these projections traverse the actual curvatures and whatnot of the white matter. And you know, if time delays are important to you in your models, as Victor has already mentioned, they are in the virtual brain, then, you know, this is really nice to have this kind of information for the models, right? But for tractography, I think most people are probably already aware that there are lots of disadvantages to tractography and limitations that it has. And one limitation is that it can be quite inaccurate. So I'll show you an example here. On the left are a couple of examples of synthetic data that are considered to be ground truth anatomical white matter tracks. They're synthetic reconstructions of the major tracks of the human white matter bundles. On the right three columns are three examples of what tractography algorithms are able to reconstruct of these synthetic data. And you can see that it can really range. So in deterministic tractography algorithms, they tend to underestimate connectivity. So you see that especially for example, in this case, the connectivity that's reconstructed can be very sparse. It's missing a lot of the existing connections. In probabilistic tractography, you have the exact opposite problem where you tend to be able to recreate most of the existing connections, but you also get too much. You get a lot of false positives in the reconstructions, right? And then on top of its inaccuracy, tractography can be really susceptible to choices in the parameters that you give the algorithm. So how much each step is allowed to take a turn and so on, and as an example. So when we came to building the virtual brain for, the virtual macaque brain, we took a best of both worlds approach in which we, for the purposes of having a connectome that is accurate, directed, and cover the whole macaque cortex, we used data that we had from tracer studies. And then to enhance that tracer connectome, we took tractography data that we had from imaging studies, where we estimated the weights and the track lengths of those connections. So this is, I admit, is a very busy figure. I don't really want you to see this too much in detail. I just wanted to describe the workflow kind of holistically of the macaque, the TVB macaque, and how we reconstructed it. And in the next few slides, I'm going to work through the pieces of this workflow with you. So the first step that we took in creating the virtual macaque brain was optimizing at the individual level of each animal, each macaque data set that we had, a subset of connections for tractography. And then once we determine for each animal their optimal tractography settings, we apply that those settings to tractography across the whole brain, sorry, the whole cortex. From there, we combine the tracer and tractography data to create the TVB Macaque package, which Jennifer already very nicely showed you an example of what you can do with it. And then in the end, I'll show you another example that we have of a resting state fMRI simulation using the TVB Macaque. So that very first step where we're optimizing the tractography for every animal in our data set was really designed to address the susceptibility of tractography to the choices we would make in setting the parameters for the tractography algorithms. And what we have available to us is a very nice set of tracer data where we know the weights of this particular set of connections. And this is available from Markov and colleagues on corenets.org. They provide a weighted matrix of about 91 regions, and 29 of those regions we have a full idea of what the connections are in both directions for that for those subset of connections. So this is what we call an edge complete connectivity matrix. So it's really only a subset of all of the cortical connections in the macaque, but we can leverage this and use this as our optimization target for tractography. Okay, and so what we did was try to get our tractography parameters set such that it would best mimic this optimal connectome, right? And so in our diffusion weighted imaging data were from 9 macaques. We ran tractography by systematically varying the curvature threshold, so how much at each step of the tractography the algorithm can bend, sorry, the streamlines can bend. We tried turning on and off the option of distance correction, because a lot of these tractography algorithms try to correct for the fact that it gets harder and harder to trace connections that as they increase in length. And then at the end, once we generate all the tractography algorithms, we also varied how many connections we discarded. So a lot of the time, people try to account for all the false positives that tractography may produce. And this is particular to especially probabilistic tractography, which I should mention is what we're using. And so, they assume some proportion of the weakest tracks are probably false positives, and so they discard them and throw them away. And so we vary how much we discard as well. And so again, on the left here is the target, this optimization target. This is what we're trying to recreate for each animal that we have using the neuroimaging that we have. On the right here is one example monkey. So this heat map shows how good each parameter set was at recreating this tracer matrix. And so on the y-axis is the varying curvature thresholds that we use, and also whether or not we use distance correction. The color of the the red and blue bars here gives us is the area under the ROC curve, which was the metric we used for how well matched the tractography was to the tracer data. You can see here on the right 4 examples from this one monkey of the best example, which is panel C here, and then some much poorer examples. And you can see, depending on how you set your tractography settings, you can get some very sparse results, or you can get some incredibly dense connectomes as well. Okay. So we did this for every single subject. These maps look slightly different for every one of our 9 monkeys. And we selected the best one. So in this monkey it was example C here. And for all of those best tractography connectomes, we kept only the edges that were described in the tracer data. So we did not want any false positives, but we averaged the weights of all the true positives and true negatives. Well, there were no weights for the true negatives. So here I just wanted to show you how realistic those weights probably were. And so tracer weights were described in units of the fraction of labeled neurons. So this is essentially a count of the axons that project from one region to another. On the y-axis is the tractography weights. This is really the probability of any streamline that we're seeding from one ROI to another, what the probability is to reach that ROI. And you can see that it actually gives a fairly good estimate of the connection weights, right? And of course that drops off with distance. So, you know, the best correlation between the weights is at the shortest distances, but even at like longer distances, those correlations were still significant. All right, so now that we've determined for every single individual, their optimal set of settings for tractography, we then reran all of that tractography for the whole cortex in those animals. And so we selected a whole cortex parcellation called the regional map parcellation. And we selected it because it has a very nice set of regions that are, it covers the whole cortex, which is actually already very rare, I would say, for a lot of macaque parcellations. And then the reason I really like it is that there is a matching tracer connectome that describes connectivity not just within the hemispheres, which is what these two quadrants are. So they're more densely connected because it's intrahemispheric connections, but there's also a description of the cross-hemispheric connections as well. So this is truly a whole cortex tracer connectome. And by applying this parcellation to the tractography data that we have, what we're doing is we're trying to get an estimate of the weights for this particular parcellation, so we can try to address the nearly binary nature of this tracer connectome, right? And although there are, I guess, categories of weights in this connectome, it's really only three categories: is strong, moderate, or weak. And we know the weights in the tracer data, so the number of axons that traverse any cortical-cortical connections, can span a range of about 6 magnitudes, so 10 to the minus 6. So these are just some examples of what the tractography connectomes, when we repeated the tractography across animals, you can see there's actually quite a bit of variability to, even if, you know, these are optimized to the tracer data, there's quite a bit of variability between animals. Those are the connection weights that are estimated in red. In blue are the track lengths. Okay, and so all we do in the next step is keep the tracer edges, so keep all the connections that are existing in the tracer data, and just apply the average tractography weights and lengths to create what essentially is that connectome that Jennifer used in that simulation. And so there's a weights matrix in this package for TVB Macaque and a track lengths matrix. And just here in panel C, I'm showing you that those weights have, they follow a log normal distribution, which is actually quite similar in shape to what we know from tracer data. So in red here is the distribution of weights from tractography. In black is the distribution of weights from tracer data. And then on the bottom is just showing that, you know, the track lengths make some sense from our tractography algorithms, that, you know, they're correlated fairly nicely with the Euclidean distance between any two regions, and that the interhemispheric track lengths, the ones that cross hemispheres, are longer in general, for example, than the intrahemispheric ones. So I think Jennifer already showed you the package that's available from the TVB-data repository on Zenodo. There's another one here as well, where we include the weights matrix, the track lengths matrix. It also includes the regional map parcellation image file in case you want to use that parcellation ROI mask for your own data. If you have neuroimaging data, for example, there's the lookup table for the region labels, and there's also a cortical surface file. So if you're interested in doing any kind of TVB modeling at the level of the surface, then the mesh files, so like the faces, the vertices, and the corresponding labels are a part of this package as well. Okay. So next I want to just show you very simply one of the example resting state fMRI simulations we've done with this TVB Macaque package. In this case, we applied a reduced one-one neural mass model to each node, and there are 82 nodes in this particular connectome. Those models of neural masses are coupled excitatory and inhibitory populations that are then all coupled together based on that, the description of the connectome, with a global coupling parameter G, scaled by the global coupling parameter G, and also there's this other parameter sigma that is noise that's injected into the excitatory populations. Right? So the particular TVB version that we use here is called fast TVB, and it's fast TVB. It's called fast because it's implemented in C code, and it's available here if you are so inclined to play with it. So running this resting state, this is a resting state fMRI simulation. So we generate the population activity for each ROI, but then we also then apply the balloon model to get the hemodynamic responses, so bold signals. Using those bold time series, we then calculate functional connectivity simply with a Pearson correlation. And that's shown here as an example. And then we did a simple parameter optimization where we varied the global coupling G and sigma, the noise parameter, in a fitting scheme to see what pairing of G and sigma gave us the best fit to empirical functional connectivity that we computed from the resting state fMRI data from those same 9 macaques. So here is a heat map, I guess I could say, of the similarity between the empirical and the simulated functional connectivity across the various iterations of global coupling and noise that we tested. Here in the asterisk is the point at which the cosine similarity between the empirical and simulated data was maximal. This is the functional connectivity matrix at that point. And you can see it bears some resemblance to the empirical functional connectivity. This is the average across the 9 macaques. And so what we did was then take the simulated time series from the TVB model and extracted the first 3 principal components just to have a look and see if they made some sense in terms of resting state networks, and they seem to. So what we were able to extract from those first 3 components was something that looked like a frontal network, so that's shown here in the red, a superior temporal network that's shown here in the green, and a visual network that's shown here in the blue. So I wanted to leave you some resources before I finish. There is a tutorial on the TVB website that if you are looking to start doing some TVB macaque simulations, you should check out. It's part of the TVB EduPack, and so you'll find at the top left of the website when you go to virtualbrain.org a link to the EduPack, and in there are a number of over 40 tutorials now. One of them is called Modeling a Virtual Macaque Brain. It was a 1-hour workshop tutorial that was put on, led by Julie Cordial, and I think she's in the audience today, where she walks through how to set up the connectivity. And here is actually a really nice screen grab of the phase plane visualization that Victor said I mentioned in his talk, and then about how you can follow kind of the trajectories of the dynamics of your model using these visualizations. And so I encourage you to go watch that if you'd like to start modeling the macaque brain in TVB. Julie also put together a really nice notebook, kind of replicating actually the simulations that I just showed, and that's available on her GitHub here. And again, she She walks through how to load the connectome into the Jupyter notebook and how to set up the model, the reduced Wang model that we use in the paper, and then visualizing, simulating and visualizing all the results. Okay, and then I wanted to leave one last little resource in case anyone is interested in particularly for primate neuroimaging. The neuroimaging data that went into TVB Macaque are also openly available on openneuro.org, and that includes both the raw and pre-processed anatomical, resting state fMRI, and diffusion weighted data that we used to build this package. And of course, many of you probably already know about these two really nice initiatives that have come online fairly recently, is the Primate Data Exchange for neuroimaging data, and also the Primate Resource Exchange. So both of these are geared specifically for sharing not just data, but tools for primate neuroimaging. So it covers not just macaques, but marmosets as well. And I think to some degree, chimpanzee neuroimaging. And so, and these tools really help kind of address some of the challenges that are unique to dealing with neuroimaging data in primates, and non-human primates, I should say. And then I'll finish with what we have planned next for TVB and the non-human primates. So the first is an extension of the virtual macaque brain, which is into a stroke model. We have from our collaborators a really nice set of neuroimaging, single neural recording, and behavioral data from a few macaques that have had parts of either FEF and or DLPFC, so dorsal prefrontal cortex lesion with endothelin 1, which induces vascular well, it induces ischemia, so it mimics very nicely a stroke. And so there are data there that are pre-lesion, and then they follow up on multiple time points up to 16 weeks post-lesion. And so I'm in the process of building that now. And then I think this has been mentioned several times already today in terms of what's being shown in marmoset and different levels of data. So I've also started to build the virtual marmoset brain, and there is this marmoset brain connectivity atlas at marmosetbrain.org, and it's a really nice set of weighted tracer data for the marmoset, and they have for some subset of these regions a edge complete matrix. That means in combination with marmoset imaging data that we have from our collaborators, and specifically diffusion weighted imaging data, and there's an example here of how they are able to trace and do tractography in marmosets at high field MRI. What my plan is, and I've been starting to do, is to build the virtual marmoset brain in the exact same way that we've done for the macaque. Right? I'm really excited to see where those go, and especially with all the emerging data now from the marmoset, I think this would be a really useful tool to have. Okay, so I end by thanking all my collaborators across many different institutions, and also I want to put in a shameless plug for a special issue that I'm co-editing in Frontiers on the neuroinformatics of large-scale brain modeling. So for many of you, I think you have some interesting work that might fit very well with this particular topic. So please consider submitting your papers to that. All right, thank you. I'll end there and happy to take questions.